Hello, good morning, and welcome to my class. I'm Momita. I'm a PhD in bioscience. I've got my PhD from Kent State University, Ohio. Thereafter, I came back to India. I worked in the pharma sector for some time, and thereafter, I left the pharma sector and I joined back into the education sector because I love mentoring and teaching. And so I have been mentoring and teaching students ever since. Thank you for being in my class today, for being with me today. So, today I'm going to take you through a lecture on mitochondria. Now, mitochondria is also called the powerhouse of the cell. So, that's a pretty interesting name. So, we will learn all about the mitochondria and we will try and understand why it is called the powerhouse of the cell, what is so interesting about it, what are its structure and functions, and so on. So let's get started. Now, what are our learning objectives in this class? Which means at the end of the class, what will you get to know? As I said, you will get to know what is mitochondria, then why we should study about it. Why is it important? So, whenever you study any topic, always ask this question: why am I studying this? Why is it important? Okay. And what are its structure and function? So whenever you are studying some object, you should be aware of its structure and function. These are the two most important elements. And what if it goes wrong? So, to understand the function of one thing, you would need to understand how do we get to know that what is of the function of a certain object. So we try to understand, what if it stops working, what if it goes wrong, then what all happens. So in most cases, from this we get to know what are all the functions. Okay, so this we call malfunction. So when something goes wrong, when something doesn't function properly, it's called malfunction. Okay, so we'll understand the malfunctioning of it also. Okay, now let's come to what is mitochondria. So you probably know this, that mitochondria is a cell organelle, which means cell is considered to be the smallest subunit of any living organism. Now, within the cell contains some things, right? It is a living thing. So, the cell contains certain objects inside it, which are known as the organelles. These each have their own functions, and taken together, these organelles help the overall cell to perform all its functions, life functions. So, mitochondria is one such organelle. Now, to give you an idea of how tiny it is, so see, it is 3 to 10 micrometer in length. It varies, the size varies. So, by 3 to 10, I mean the size varies. So, this is the variation in length, and zero point 0.2 to 1 micrometer in diameter. So, you can imagine it is measured in micrometer. It's microscopic. Okay, so it is very, very small, but small, but you cannot really underestimate it. And not only can you not underestimate it, it performs some of the most important functions inside the cell. We'll get to see later. Now, what is its shape? It is oval, rod shaped, usually. But again, its shape may also vary. Nothing in this living world can be really determined or fixed to one shape, to one size. Certain variability exists in life, in all cases. And that is what makes life so interesting. So, the mitochondria is no exception. Now, mitochondria is present in most cells except for prokaryotic cells. So I suppose you already know what prokaryotic cells are. So prokaryotic cells are the primitive cells which do not have an organized nucleus. So they have a nucleoid. So that is another topic. I'm not going to delve into it. Now, even though I say primitive cells, prokaryotic cells still exist in the form of bacteria, cyanobacteria, and all that. So we'll get to it later. Now, in humans, most cells contain mitochondria except for the RBCs. So, RBCs have gotten rid of most things except a few things inside their cells. because they want to optimize their surface area for absorption of oxygen. Mitochondria was first observed by Golliker, and it was named mitochondria by C. Benda. So these are just facts that you need to remember, okay? And I just thought of mentioning those. Now, coming to the most important part, I find this the most important part. Whenever I'm looking at things, whenever I'm studying things, the first question I ask is, why should I study it? Why is it interesting? Right? So it has to be interesting in some way because, it is prevailing all over our biology. So there are, in fact, laboratories that study mitochondria itself. So it has to be quite important. It has to be very interesting. Now, mitochondria is interesting for various reasons. The most dominant reasons out of them are these two. So, first is it is semi autonomous. Now, what is meant by semi autonomous? This means that it is almost independent. Autonomous means independent, something that can independently function. So, mitochondria cannot completely independently function. If you take it out of the cell, it will not be able to perform all the functions or survive, but it can perform its function to a great extent. Okay, why? Because it has its own DNA, circular DNA, and some mRNA and ribosomes, with the help of which it can synthesize some of its proteins. Now look at this word, some proteins, not all proteins. That is why it is semi-autonomous. And this DNA was first observed by Nass and Nass. Now, isn't it interesting that no other cell organelle has a DNA? Now, of course, we are not talking about the chloroplast, that is another story. But in general, other cell organelles or most cell organelles do not have this DNA. They do not have the capability to synthesize their own proteins. Now, mitochondria do not synthesize all their proteins, true, but at least it can to some extent, right? So that makes it very interesting. And why is it so? Why is it structured like that? It also has a double membrane. Most cell organelles have a single membrane. So why does mitochondria differ from most other cell organelles? Because the theory is that mitochondria is an endosymbiont. What is meant by endosymbiont? Which means long back, when eukaryotic cells had diverged from prokaryotic cells during evolution, it so happened that some eukaryotic cells probably ingested, right? You can see it is ingesting. So it had ingested some of the any one of the prokaryotic cells. I'm going to move myself so you're able to see the whole endosymbiosis. So see, maybe this was a prokaryotic cell hanging around and this was the eukaryotic cell. So slowly, what happened is, maybe by chance, it just ingested the prokaryotic cell. And ever since, the prokaryotic cells stayed inside, right? And through evolution, they co-evolved. So probably because through this process of symbiosis. So the prokaryotic cell said, I'm going to provide you energy. We'll come to the functions later, but just as a precursor to it. So the prokaryotic cell said, I'm going to provide you all the energy. You provide me with all the other necessary materials that is needed for me to survive. And also you provide me with protection, right? So eukaryotic cells are so well organized, it would be greatly protected if it's inside the eukaryotic cell. So naturally, there was this symbiotic relationship, and hence it is known as endosymbiotic because it took in the prokaryote through, this process of uh, endocytosis, and then this remained to be a symbiotic relationship. So that is what is meant by endosymbiotic. Okay. So now let me try to move to the next slide. Okay. Right. Okay. So So, coming to the fun facts around mitochondria, because we always like to start with this, it makes us really interested, right? Okay. So, coming to the five fun facts, the lifespan of mitochondria, each mitochondria around 5 days, and then it regenerates, it divides, right? Divides to form more mitochondria. So, don't be scared, all your mitochondria are not vanishing. It is slightly yellowish in color because it has this pigment called riboflavin. Its DNA is rich in GC. So we know that DNA has bases, four bases, right? A, T, G, C. So it is GC rich, right? Guanosine, cytosine. 70% of total cellular enzymes are found in mitochondria. Why so? You should give it a thought. Why are so many enzymes concentrated in the mitochondria? This means it carries on a lot of metabolic reactions, right? Mitochondria are fewer in plant cells than in animal cells. If you understand its functions, you will understand why. Because animal cells have a lot of movement to do. Plant cells also move, but to a lesser extent, right? They don't move from one place to another. They show movements like phototropism, but that does not require as much energy as animal cells do. So it has to do something with the energy. So we'll get to that again. Now I'm gonna move myself again so that you can see the rest of the sentence here. Okay. So yeast cells, okay, yeasts are, they are single celled, right? So they have one giant mitochondria. But what do we have? Each cell has around 500 to 800 mitochondria, but the number varies depending upon the function of the cell. So mitochondria has one very important function which we will get to, and that determines everything that you see here. The number of mitochondria that are there. Okay? And then mainly the number of mitochondria that you need in a cell. Okay, so that is dependent on the function. Okay, let me try to go to the next slide. So now, as I said, structure and function. These are some of the very important things here. Okay, so let's understand its structure. So as I said, mitochondria is double membrane bound. Always remember that because this is something that is very peculiar to mitochondria. Okay, not all the other cell organelles are double membrane bound. Most are single membrane bound. Mitochondria is different. Why? We already know that. Now, naturally, if there are two membranes, there will be an outer membrane and there will be an inner membrane. Now, inner membrane here is extremely important. It is folded into finger-like projections like this. Okay? So this. So these are called cristae. And these cristae have tiny little structures on them, lots of them. Those are called oxysomes or F1 particles. These play a very important role in its function. We'll get to see how later. And then there's this outer membrane. So outer membrane is made up of some of the regular membrane stuff like phosphatidyl inositol, cholesterol, cardiolipin, subunits of Parson, etc. etc. Okay. Now subunits of Parson are similar to F1 particles. Okay. So they also contain these to some extent. So they must be important, right? So we'll see how they are important. On to the next slide. Look at these F1 particles. First, I wanted to take a look at these F1 particles. This is a very, this term is very new to you, right? So you should know what is an F1 particle. Why are they there all over the mitochondrial membrane, especially the inner membrane? Well, these are particles like this. They have three portions. They have this head, the stalk, and the base. So the head is called F1, stalk is called stalk, and the base is called F0. So I hope now you know the three parts of the oxysomes, right? What is their function? They function as ATP synthetase. And look at this. They catalyze ATP synthesis, one of the most important functions in living organisms, right? Synthesizing energy. We cannot synthesize energy, then we'll be dead in no time. So, as I said, mitochondria performs one of the most important functions in living organisms. So, it catalyzes ATP, right? ATP synthesis. Catalyzes ATP synthesis. Yeah. So, as we know, there are these membranes, but does it stop with the membranes? No, it doesn't stop with the membranes. There's something inside, right? Inside these membranes. That is the matrix. We call it the matrix. And that contains the DNA. We have already mentioned that the circular DNA, the 70S ribosomes, the enzymes, cofactors, inorganic ions, organic molecules, other proteins. So it is just a combination of so many other things that the mitochondria needs to survive, right? So as I said, mitochondria has 70S ribosomes. Did you notice that? Who has 78S ribosomes? Well, yes, you are right. Prokaryotes have 70S ribosomes. Eukaryotes, they do not have 70S ribosomes, and I leave it up to you to find out what kind of ribosomes do eukaryotes have. So the fact that this is a double membrane bound organelle with 70S ribosomes and that is semi-autonomous itself clearly indicates that it is an endosymbiont, right? There is an exception though. Mammalian mitochondria, it has 55S ribosomes. Some sort of evolution it seems. Okay? So remember this part, okay? Because this is a peculiarity again, and we almost always tend to remember the peculiarities, right? Okay, getting back to functions. So now we know the structure. Two membranes, inside there's a matrix, and there are these oxysomes all over the inner membrane. Now let's come to the functions. So as I said, these F1 particles are there producing ATPs. We already know, it has been revealed that it produces most of the cellular ATP. Now cellular ATP is produced by one major reaction which is known as the Krebs cycle. We'll get to all of that later. This is not within the scope of this lecture. But let us say that this Krebs cycle is occurring in the matrix of the mitochondria. Okay? And the inner membrane, inner membrane also has cytochromes. Okay? They carry the other part of ATP synthesis, which is oxidative phosphorylation. So ATP synthesis is not a one-step process. It is a multi-step process. So there's glycolysis, there's Krebs cycle, there's oxidative photophosphorylation, and then there is this proton pump, whole proton pump gradient, which leads to ATP synthesis. So this proton pump is maintained by the oxysomes that we saw earlier. Okay? We'll get to know all about it in more detail, but these are the basics. So the proton pump, this oxysome serve as a proton pump which creates a proton gradient through which ATP is synthesized. The inner membrane undergoes oxidative or carries out oxidative photophosphorylation because it has cytochromes, and the matrix that carries out the Krebs cycle. So most of this energy production, the process of energy production occurs within the mitochondria. Hence, it is known as the powerhouse of the cell. It gives us power. Energy gives us power, right? Hence, animal cells have more mitochondria than plant cells because we need more power. We have to move around all the time, right? So now everything is falling in place. Okay, so as a result, as I said, the number correlates with the cell activity. Even within our body, the number of mitochondria vary from one cell to another, depending on the activity of the cell. So muscle cells will have more mitochondria, right? Because they have to perform greater power related functions, right? So they will have more mitochondria. So now that you know its function, you would be able to predict which kind of cells will have more mitochondria. And we already know that RBCs do not have mitochondria, so leave it out of the equation. But mitochondria is not. So mitochondria is not only providing us with power, but it is also the site for many other reactions, mainly synthesis reactions. So we saw ATP synthesis. Other than that, other synthesis reactions also occur, like synthesis of heme protein, cytochrome, myoglobin, very important molecules. So, as I said earlier, mitochondria is extremely important. Hence, it is studied as a subject in many laboratories across the world. So, recap. Okay, I always do a recap, a small recap after going through a few modules on the lesson. Okay. So, recap is outer membrane. We know mitochondria is double membraned. Outer membrane, it is permeable to small molecules, giving you some additional information also. Inner membrane, it is a site of electron transport chain or oxidative phosphorylation. So always remember this, okay? Oxidative phosphorylation is also known as electron transport chain. Then the inner membrane is folded into finger-like projections called cristae, which contains the oxysomes, remember. The matrix, it contains enzymes, mitochondrial DNA, and it also carries out the Krebs cycle. And then there is this intermembrane. So naturally, when there are two membranes, there is a space between the two membranes. This intermembrane space is the area between the membranes. Now, when you study the proton pumping and proton gradient and all that, you will understand the significance of this intermembrane space. But I just wanted to mention this in addition so that you have that in mind. Okay, the two membranes are not pasted together. Have some space in between, that is the intermembrane space. A mitochondria is majorly involved in energy production. It performs cellular respiration. It breaks down glucose to produce ATP. It uses the proton gradient, which I already mentioned before, to generate energy. And it takes the help of oxysomes or the F1 particles or F0 F1 particles for this. This helps in creating this proton gradient. Now, you must be curious now, very curious about what is this proton gradient? Keep on talking about. It's very important, but we will deal with it as a separate topic. So the total amount of energy that the mitochondria produces is around 36 to 38 ATP molecules per glucose molecule. So the starting point is glucose molecule. So we derive the energy through a series of reactions that these glucose molecules go through to produce like 36 to 38 ATP molecules. And it houses the Krebs cycle. So Krebs cycle is the part of energy production only. Okay? So these are some of the things to jog your memory again. We have discussed all of this. I keep reiterating so that it gets into your brain some of these key terminologies. Okay? So that later on, when we get into these reactions, you will be able to relate. Oh, okay, so this is taking part in the matrix of the mitochondria, connecting the dots. Very important. So I always encourage you to connect dots, okay? So not read chapters as discrete entities, but try to connect one thing with the other. So when we do bioenergetics later, try to connect it back to the mitochondria lecture, okay? So there are many other important functions. So synthesis is one aspect of mitochondria, and it is it's a major aspect. Other functions include calcium ion storage, regulation, cell signaling, cell death, apoptosis, again, another topic that we need to deal with in detail later. Apoptosis regulation. Apoptosis means cell death, basically. Heat production, steroid hormone synthesis. So you can imagine the reason why I listed these is to make you understand how important mitochondria is. Again, as I said, when you have to understand the significance, the impact of something, you have to understand what happens if it's not functioning properly, malfunction, right? So mitochondrial malfunction can affect any organ system. Okay, any organ system, it can be really bad. It is usually maternal inheritance. The mitochondria that we have is inherited from our mother. Okay, most other organelles, they are a combination of both parents. But mitochondrial inheritance is mainly maternal in nature. Another interesting fact, right? So I keep throwing in these interesting facts so that you don't fall asleep. No, I'm just joking. Even I like interesting facts here and there. Okay. Now, again, coming back to malfunction. So, mitochondrial malfunction causes muscle weakness. Okay. So, this is known as mitochondrial myopathy. So, as I said, muscles have a lot of mitochondria naturally. It's intuitive, right? And any problem with the mitochondria in the muscles causes mitochondrial myopathy. It can even cause a combination of muscular and neurological problems, which is known as mitochondrial encephalomyopathy. Okay, encephalo because of the neurological component. There are many other conditions that can be caused by mitochondrial malfunction, like impaired vision, hearing loss, abnormal heartbeat. Just imagine. So, heart, brain, muscles, most important organs are powered by this powerhouse. Okay. It can even cause diabetes and stunted growth. So, just imagine how important mitochondria is to us. Moving on. So, here's a summary again. Mitochondria is a double membrane cell organelle, inner membrane folded into cristae containing oxysomes. It is an endosymbiont, it is semi-autonomous, it is the powerhouse of the cell, it serves as a site for both Krebs cycle and oxidative phosphorylation, Krebs cycle in the matrix, oxidative phosphorylation or electron transport chain in the inner membrane because of the presence of oxysomes. That is the proton pump because of the presence of oxysomes. And it performs a plethora of other functions. And disorders, as we see, I'll clear out this area, I'll move myself again. So, disorders lead to muscle weakness and other related symptoms. So, as we see, we have covered a bit and only a bit of mitochondria. That too, not the entire thing. We have so many reactions to understand, right? So we'll do all of that bit by bit. This was just an introduction. I hope you understood all that. Now, after every class, I always give you some points to think about. It's good to, stop and think about a few things, ruminate on a few things that helps you develop deeper concepts. Okay? So this is not just for memorizing. It is also for thinking. You should develop your critical thinking skills. So think about this. I said mitochondria, not me, but it has been shown scientifically that mitochondria is an endosymbiont. What kind of bacteria do you think this endosymbiont was before it was being ingested? So we know that bacteria are of different categories: aerobic, anaerobic, facultative aerobes, facultative anaerobes, obligate aerobes, obligate anaerobes. What do you think this mitochondria was, depending on what its functions are now, right? So think about that. Think about why this entire endosymbiosis process stuck on. Why was it so successful? As I said, through evolution, these two paired up and they remained paired. Why do you think it was so successful? It's easy, right? When you come to think of it, we have already covered these points in the lecture. And which cells? Now, I've already covered this to some extent, but you might also want to find out. I just mentioned muscles on purpose because I want to find out which other cells are likely to have a higher mitochondrial count. Okay, so these are some of the things that you can think of. Thank you so much for listening to the lecture. Okay, so I hope we will get to meet again, and I would love to talk about the further details on the various concepts that I just glazed over today. Hope to see you soon in my class. Hope you enjoyed